Imagine a world where we could flick a molecular switch to block viruses like herpes from sneaking into our cells and sparking nasty illnesses. That's the thrilling promise of groundbreaking research out of Washington State University, and it's got the potential to revolutionize antiviral treatments. But here's where it gets controversial. By tweaking a single protein interaction, scientists are essentially outsmarting these crafty invaders, raising questions about the ethics of manipulating nature at such a fundamental level. Could this lead to over-reliance on tech-driven solutions, or is it a game-changer for public health? Stay tuned as we dive into the details. In a study published in the journal Nanoscale, accessible at https://pubs.rec.org/en/20five/d5nr03235k, researchers from the School of Mechanical and Materials Engineering and the Department of Veterinary Microbiology and Pathology at Washington State University, located in Pullman, Washington, uncovered a clever method to interfere with a key virus protein. This approach could prevent viruses from infiltrating cells, where they wreak havoc by causing diseases. For beginners, think of viruses as tiny, sneaky burglars that need to pick locks on our cell doors to break in, and scientists just found a way to jam one of those locks shut. Viruses are incredibly clever, as explained by Jin Liu, the lead researcher and a professor in the School of Mechanical and Materials Engineering. The entire invasion process is incredibly intricate, involving numerous interactions, Liu said. Many of these might just be distractions, like background noise, but a few are absolutely vital. By focusing on these critical points, the team targeted a fusion protein, a molecular tool herpes viruses use to merge with and penetrate cell membranes, leading to a range of health issues from cold sores to more severe conditions. One major hurdle in fighting herpes viruses is our limited grasp of how this fusion protein unfolds and breaches cells, which is why effective vaccines remain elusive. To address this, Professors Prashanta Dutta and Jin Liu from Mechanical and Materials Engineering harnessed artificial intelligence and molecular scale simulations. They combed through countless potential interactions among amino acids, the basic components that build proteins, to pinpoint a crucial amino acid enabling viral entry. Using a custom algorithm, they analyzed thousands of these interactions, and then applied machine learning techniques to sort out the noise and highlight the most impactful ones. This is like using a high-tech detective to sift through a mountain of clues and zero in on the smoking gun. Under the guidance of Anthony Nicola from the Department of Veterinary Microbiology and Pathology, the team engineered a mutation in this key amino acid. The result? The herpes virus couldn't fuse with cells anymore, effectively halting its ability to cause infection. For context, imagine trying to open a locked door with a jammed key. The virus just couldn't get a foothold. The simulations and machine learning were game changers here, Liu noted, because testing a single interaction experimentally could drag on for months. Out of thousands of possibilities, we isolated just one critical interaction, Liu remarked. Without simulations, we'd be stuck in trial and error mode, potentially wasting years. Blending computational theory with hands-on experiments is a powerhouse for speeding up discoveries in biology. And this is the part most people miss. While they've confirmed the importance of this interaction, the researchers still lack a full view of how mutating it ripples through the entire protein structure. They are optimistic about deploying more advanced simulations and machine learning to map out the bigger picture. There's a disconnect between what lab tests reveal and our simulation insights, Liu admitted. Our upcoming challenge is understanding how this tiny tweak triggers larger-scale structural shifts. It's no small feat. The project team included PhD students Ryan Odstasil, Albina Marchio, and McKenna Hull, alongside Liu, Dutta, and Nicola. Funding came from the National Institutes of Health, underscoring the public health stakes. This research isn't just about science, it's stirring debate on the future of medicine. For instance, some might argue that relying on mutations to block viruses could inadvertently create super-resistant strains, forcing us into an arms race with nature. Others see it as a beacon of hope for diseases we've long struggled against. What do you think? Is this the dawn of a new era for antiviral therapies, or are we playing with fire by meddling at the molecular level? Do you believe this could pave the way for herpes vaccines sooner than we expect? Share your opinions and counterpoints in the comments. I'd love to hear your take.